All right, let's dive into the pretty incredible journey of a single bag of donated blood. It all starts with a simple, generous act. But what happens next? Well, that's where we enter this hidden world of high-stakes science, a world of unbelievable precision and life-saving skill.
Think about it. You know the drill, right? You get the little pinch, watch the bag fill up, grab your juice and cookies, and you're on your way. But for that bag of blood, its adventure is just getting started. So let's follow it.
And this isn't just about sticking it in a fridge. Nope, this is about unlocking all the biological secrets packed inside every single drop of that donation, all to make sure it can safely give someone else a second chance at life.
But before any of that lab magic can happen, the journey really begins with the donation itself. And this very first step is surrounded by some super strict rules, all for one really important reason: safety.
So what's with this number? 13 grams per deciliter. For a male donor, that's the absolute minimum hemoglobin level you need to have. It's one of the very first safety checks they do, and it's critical. It makes sure that you, the donor, can give blood without putting your own health on the line. For female donors, by the way, it's just a little bit lower, at 12.5.
And believe me, that's just the tip of the iceberg. There's a whole checklist of vitals they run through. Your weight, your pulse, your blood pressure, your hematocrit, every single one has to be in a very specific, healthy range. It's a serious screening process. And this quote from someone who works in a blood bank really just says it all, doesn't it? The whole point is to protect everyone, to help a patient not accidentally create a new one.
Okay, so the blood has been donated, it's past that first test. Now we go behind the scenes. We're stepping through those closed doors and into the world of the clinical laboratory. This is where the real detective work begins.
This place is the domain of the unsung heroes of medicine, the clinical laboratory scientists. These are highly trained professionals working in an environment where there is literally zero room for error. And their job starts before they even lay a finger on a blood sample. Just look at this protocol. Every single step, from how long you have to wash your hands to the exact order you put on your gear, it's all designed to stop contamination, to protect the sample, and to keep the scientists safe. It's like a ritual of pure precision.
Okay, safety protocols are locked in. Now the scientists can get to their real mission, decoding the blood. And this is where it gets really fascinating, because we're going way beyond just, are you type A or type B?
Yeah, this might look like some crazy hard genetics exam question, right? Well, that's because it is. This is an actual practice question for the people getting certified as lab scientists. This is the kind of mind-bending puzzle they solve every single day to figure out a person's exact genetic blood type.
So to give you a taste of just how complex this gets, let's talk about one system you've probably never heard of, the Lewis system. See, unlike the A and B antigens we all know about, which are actually part of the red blood cell, Lewis antigens are made somewhere else in your body, and then they just stick to the outside of the cell. And knowing this is so important because some of these antibodies can cause some really nasty, unexpected transfusion reactions.
And figuring out your Lewis type isn't straightforward. It's this whole genetic dance between at least 3 different genes. Are you a secretor? Do you have the Lewis gene, the H gene? A lab scientist has to understand how all of these things interact just to get the blood type right and avoid any problems.
So, you're probably wondering, how on earth do they see all this stuff? It's not like they're just peeking through a regular microscope. No, they have a whole arsenal of incredibly sophisticated technology at their command.
Take this, for example, spectrophotometry. The idea is actually pretty brilliant in its simplicity. You shine a light through a sample. The amount of light that gets soaked up by the sample tells you exactly how much of something is in there. It's used for everything from checking for damaged red cells to measuring protein levels. It's a total workhorse of the clinical lab.
And then there's electrophoresis. This is so cool. Think of it like a molecular racetrack. You use electricity to make proteins run through a gel. The little guys move faster than the big guys, so everything separates out into these neat little bands. This lets scientists spot things like abnormal hemoglobin or other weird proteins that might signal a disease.
So here's the bottom line. These amazing tools are what allow lab scientists to take a sample of blood and turn it into cold, hard, life-saving data. They make the invisible visible. They can measure pretty much everything that matters for your health and for finding a safe match.
So the blood has been donated, it's been screened, it's been analyzed down to its genetic core. But hold on, we are not done yet. Not even close. There is one last, absolutely critical test before that blood goes anywhere near a patient.
After all that work, you'd think, okay, it's good to go, right?
But what if? What if there's some super rare, undiscovered antibody hiding in the patient's own blood? Or a really uncommon marker on the donor cells? You have to do one final check to prevent a total disaster. And let me tell you, the stakes here are as high as they get. A bad match can cause an acute hemolytic reaction. That's when the patient's body immediately attacks the new blood, and it can be fatal. Or it could be a delayed reaction, where days later the patient's immune system kind of remembers an old fight and launches a whole new attack.
And that brings us to the final, ultimate safety check, the major crossmatch. The idea is so simple but so powerful. You literally take a little bit of the patient's plasma, where their antibodies live, and mix it with a little bit of the donor's red blood cells. It's the final handshake, a meeting between donor and recipient in a test tube.
And the answer you get here, it's a hard yes or a hard no. There is no middle ground. If you see any reaction at all, any clumping, any cells breaking apart, that's it. Game over. The unit is incompatible. It means the patient's plasma is attacking the donor cells right there in the tube, and it would do the exact same thing inside their body. This one simple test is the final firewall that saves lives every single day.
So after all of this, after following this whole journey from a donor's arm through this high-tech maze of a lab, all the way to that final handshake in a test tube, the next time you see one of those bags of blood, what are you going to see?
Because it's not just a bag of red liquid anymore, is it? It's a story. A story that started with the donor's generosity, continued with meticulous science and incredible technology, and was made safe by the amazing skill of clinical laboratory scientists. It's a whole hidden world that makes the gift of life possible.